OmicsOffice Suite is an application for the analysis of omics data. It runs on the Spotfire platform and is organized as a core set of components available to any OmicsOffice user. Additionally, it comes with a series of vertical workflows that can be added to this core set containing technology-specific workflows. The vertical workflows have the additional capacity to load raw input data from RNA-Seq and ChIP-Seq in the form of aligned reads as well as microarray and qPCR data. In this way, we aim to help the researcher move from raw data to biological relevant results in a smooth manner. OmicsOffice provides a much-needed bridge between the field of biology and data analytics, providing researchers with a set of specific tools and workflows that addresses their specific needs, in addition to the increase in power and productivity that results from the use of Spotfire. Here we can see a diagram with the organization of the OmicsOffice Suite, with the core elements in the upper area and the vertical workflows on the bottom. Here we can place the third-party tools, the output of which can also benefit from our software. The tools that are available in the core include data integration, functional analysis, data pre-processing, and a variety of other tools oriented for the manipulation and statistical analysis of biological data. The tools provided by OmicsOffice are both powerful and reliable, while still maintaining simplicity for the user. This can be illustrated by the click-and-go philosophy followed in our workflows, where the user can provide the minimum configuration necessary and the analysis can be run using one of our predefined workflows with a few clicks. The results are provided in an easy-to-interpret manner, taking full advantage of the flexible visualization and analysis environment of Spotfire, with quality control information that clearly highlights potential problems in the data such as poor correlations, outliers, and biases. Although strict quality control measures are important, they are not useful if unaccompanied by a reliable analysis software. As such, the algorithms and tools used in our OmicsOffice software are all peer-reviewed accepted in the scientific community, as you can see in this list. Since an example is always better than a set of slides, here we can see the process of setting up and executing RNA-Seq analysis. First, we select the required workflow and indicate the files to be analyzed and upload the relevant experimental design. In this example, you can see we loaded raw alignment files. This ability to load raw data files into Spotfire is one of the major advantages provided by our vertical workflows, as many formats commonly used in biology cannot be easily imported into Spotfire without major pre-processing steps. We can now select the workflow we need and click on the click and go button. As you can see, with a few clicks we have started the execution. The output we obtain is a Spotfire document with several pages containing different parts of the analysis. These parts are dedicated to the quality control of the input samples and helps us to visually identify if anything has gone wrong with the analysis, thereby allowing us to rapidly drill down on the source of the problem in order to decide if there is a real problem or if the warning is caused by some underlying peculiarity of the data. As the analysis has been performed within the Spotfire platform, additional downstream analysis can easily be added to the results of the workflow using both the tools integrated in Spotfire and the specific tools for biological data analysis provided by OmicsOffice. These include all the statistical tools used during the different workflow analysis steps, as well as additional tools for data integration or functional analysis. From the results of this RNA-Seq analysis, we can now go on to interactively select the set of significantly upregulated genes that we are interested in. In a few clicks, we can easily perform a gene ontology enrichment analysis, which will give us biologically relevant information on these genes that we selected. The importance of easily obtaining this type of biologically relevant information cannot be underestimated, particularly now that we are facing an unprecedented avalanche of data where optimal analysis rather than data generation is increasingly becoming the bottleneck for many an experiment. By providing clear and interactive visualizations that help to identify and communicate the specific information, we are able to help the researcher speed up this analysis process. Building on this idea of providing the researcher with tools that allow them to easily and efficiently explore their data, we have also developed the ExpressionExplorer. ExpressionExplorer can be used with both expression data analyzed with OmicsOffice workflows, as well as data analyzed with third-party tools. With ExpressionExplorer, we can easily get an overview of the data sets for which we have performed multiple comparisons over a variety of experimental variables. This allows for the easy comparison of the genes that are differentially altered over the conditions, as well as providing lists of relevant genes that are over or underexpressed, in addition to attributing important functional information to them. Once the analysis is completed, we can easily share the results in PowerPoint or in the form of an interactive web player. This allows our collaborators to further add to the data analysis and provides them with a much better feel of the results than any flat file figure can convey. In this era, in which data analysis is increasingly becoming the bottleneck of many genomic studies, we at Integromics are working hard to provide the researchers with the best tools to address this problem by bringing the data analysis and visualization powers of Spotfire into the world of biology. For further information, please visit us at www.integromics.com